Hello class, welcome to week 9. I hope that you survived the midterm. It's always a little bit rough, but after the first exam, you should have a feel for how this class works. It's not too difficult. We do a test every 4 weeks. Week 4, week 8, which is midterm, week 12, and then 16, which is the final. So this week, we're actually going over chapter 17 and 18, although I will say it's more chapter 18 emphasized. But just for the sake of covering all the material in the book, I want to brush over chapter 17 as well. So while the title of this PowerPoint is Evolution and Diversity Among the Microbes, the chapter 17 in the book is actually viruses, bacteria, and protists. So we just have 20 slides over this material. So microbes are what we call ubiquitous. They exist in all of the domains. Of course, they're microscopic organisms. They're the most abundant organism on the earth. They're very genetically diverse, and not all microbes are closely related. They live and can be found in almost every single habitat on Earth. Viruses are microbes, but they're not considered to be alive. Thus, they're not really considered to be part of any domain. So as far as microbes and us, this I think is very interesting because the total number of cells in the human body is in the trillions. If you count the microbes versus the human cells, you can see by the chart, the orange part is all the bacteria that's within our body. Which, it sounds kind of disgusting to be like we're full of bacteria, but I personally don't think it's disgusting. I think that the fact is interesting because for a long time people thought that germs equaled bad, microbes equaled bad, and that is not always the case. So microbes account for the majority of cells associated with the human body. There's 400 species of microbes in our digestive tract, so that helps us with digesting our food. We have 500 species in our mouths and about 200 on our skin. So this helps with all sorts of things like keeping things clean, generating saliva to help pre-break down our food before it goes actually into our digestive tract, protection on the outside of our skin. All those things are important. Now again, like I said a moment ago, viruses are not alive. A virus does not contain DNA or RNA, and it is able to replicate. You can just take a look at the picture down here on the way that viral replication happens. There's not any sort of metabolic activity, and it definitely requires a host cell that can metabolize because it can't do it for itself. So in step 1, the virus will attach to the host cell, and then the viral DNA copied, it uses the host cell as the machinery. So it gets inside the cell, kind of hijacks the system, and tricks it into making copies of itself. And that is how an infection happens. So the viral mRNA gets transcribed, and then the viral proteins are synthesized, and then the new viruses are released from the host cell. So it kind of gets in, hijacks the system, makes copies of itself until the body can figure out what's going on and get rid of it. But usually it takes a little while. Viruses can infect a wide range of organisms. Most viruses only affect one species or a group of closely related species, though. Some can infect multiple species, like rabies can infect any mammal and not just specific ones. And then some can also move from one species to another. That is common. And almost all influenza outbreaks are caused by a bird flu virus jumping to humans. Now, this is something that you may be familiar with recently because of the origins of COVID. They think that it started with a bat and then that jumped over to humans, which is not the first time something jumped from, we call those zoonotic diseases. So something that is carried by another species of animal that will then jump to a different host. This happens often, actually. That is why my research surrounded bats, because we wanted to look at what bacteria they held. And then on the next level, is that possible to have that spread to humans? And if so, how can we prevent it? It's just knowledge in that area is powerful because bats carry, I think the last time I looked, it was over 100 known diseases that could jump from a bat to a different animal. Yeah, so that sort of stuff is important to know. because they can cause serious health problems. Viral diseases, they can be relatively minor, or they can affect a large number of people. So you can have a pandemic, which is a worldwide epidemic of a virus, like the influenza pandemic of 1918 and 1919 that killed millions of people. DNA-based viruses are relatively stable, and vaccines can be created for those, like smallpox or polio, chickenpox, measles, mumps, etc., etc. Now, the RNA-based viruses, those mutate really easily, and they're difficult to create effective vaccines, like HIV or the flu. Flu pandemics occur about every 50 years. So what I was told one time, whenever I was looking into genetic work, is DNA is quite stable. It's easy to work with in a lab setting, but then whenever you switch over to RNA, you breathe wrong and the RNA falls apart. It's just really difficult to work with. There are scientists who spend 20, 30, or 40 years of their lives working on RNA-based research and have nothing to show for it just because it is so difficult. Now, with advances in technology, I think things are getting easier, but just comparatively, RNA is much more sensitive and harder to work with. The structure of bacteria, you can take a look at the picture here. They're very simple, actually. They have circular DNA inside, they have cytoplasm, they have a cell wall, they have the capsule for protection, the flagellum to help it move around, and then the pili, which are also like little hairs to help them move around. Very simple. They don't have a nucleus, so they'll have things to help them move around, things for a layer of protection. If you're familiar with anything like in microbiology, which you may not have gotten to yet, or you might not get to, but in microbiology, we do something called a Gram stain to find out if something is a negative or positive. If it is Gram, it's based off of the cell wall. If it doesn't have a cell wall, or like a slime coat around the cell wall, then the Gram stain can actually get to it, and it'll tell you, oh, this is positive. If it's negative, those tend to be a little bit more virulent sometimes. They're a little bit harder to treat. They might have some antibacterial or antibiotic resistance because the cell wall has to be penetrated for things to work. So you kind of find the ones that potentially in certain circumstances have a little bit more severe of an effect, have a stronger cell wall structure. It is not always the case, but Gram stains in microbiology, this is kind of based off of that structure. As far as bacterial reproduction, they reproduce asexually via binary fission. So they just make copies of themselves, and it's very, very, very fast. In less than 12 hours, a single E. coli could give rise to a population of 20 billion cells. It's ridiculous. Like whenever we go to do cultures on like a petri plate with a nutrient auger in it, you just have to incubate it for a day, maybe 2. And if you don't take it out before then, it will overgrow because they just keep replicating. They make daughter cells that are genetically identical to the parent cells. So they are exactly the same. They're just copies of themselves. We also have horizontal gene transfer. Bacteria of the same generation can exchange genetic material, and that's done in 3 ways. You have conjugation. That's where the bacterium transfers some or all of its DNA into another bacterium. You have transduction. That's where a virus which infects a bacteria transfers DNA from its previous host into the bacterial cell, so it's bringing something with it. And then you have transformation. So bacterium picks up what they call free DNA from the environment and can kind of carry that with it. Bacteria, again, they're metabolically diverse. They have a variety of ways to obtain food and energy. You have chemoorganotrophs. They feed on organic molecules. You have chemolithotrophs. Those feed on inorganic molecules. And then you have photoautotrophs. They use light energy to make their own food. So that's like photosynthesis. So you can see by the picture examples down there the different things that bacteria could eat. For example, on the photoautotrophs, cyanobacteria, that's 2.6 billion years old, and that's responsible for the accumulation of oxygen in the atmosphere. So I mean, they are beneficial, because we like oxygen. We kind of need that stuff. They can be beneficial, as I mentioned before, so it's not that bacteria equals bad. So there's good bacteria: yogurt, cheese, beer, wine, vinegar. Um, Yogurt especially has, I think it's a lactobacteria, which is it's a healthy probiotic. It's good for your guts. You need a healthy balance of bacteria in your body, in your stomach, for the body to function properly. So back before modern medicine, I believe they had something called the four balances or something similar to that, where they thought, you had parts of the body that needed to be balanced, and they were kind of on the right track. But if you went in and your stomach was hurting, then your gut was out of balance, and you had to try to figure that out. That is absolutely true. So gut health is linked to a number of health diseases. It's even linked to depression, anxiety. It's linked to a lot of autoimmune diseases. You should check it out. You should look it up. I always encourage extra research. Don't just take my word for it. Do a quick Google search and type in something like gut health linked to disease and see what you can find. Whenever I was in my evolution class, we talked about this at some length. It's extremely interesting to see how diets have changed over the years on what food's available at different times of the year, what people as a society choose to consume, and the effect that has on health, mental health, just like collectively. It's a very interesting fact. But some bacteria can help prevent diseases. The good bacteria in your body consumes all the resources, and that kind of keeps the bad bacteria out, doesn't give them resources to grow. And like I said, it helps your digestive system function. Antibiotics can kill your typical bacterial colonies. That's why if you've ever had to take antibiotics, a lot of the doctors will suggest taking your antibiotic with yogurt in the morning. You have to put the good bacteria back into your body because antibiotics are killing it all. They're killing everything, bad and good. So that can result in digestive issues or yeast infections. Things just don't function correctly if you have heavy antibiotics. And not everybody has these issues, or maybe doesn't notice these issues, but I am definitely one who I can just tell things aren't quite right if I don't eat yogurt whenever I have antibiotics, which I don't very often, but I recently just had a sinus infection, had to have 10 days of antibiotics to get over that, and I ate yogurt twice a day, just because if I don't, I'll not feel well every time I eat. Anyway, like I said, bacteria causes a lot of diseases. So you can have pathogenic, that's technically disease-causing bacteria that kills all millions of people annually. Some bacteria are always pathogenic. Cholera, the plague, tuberculosis, leprosy, etc. Some bacteria are generally harmless but can become pathogenic, like strep throat, scarlet fever, or acne. I think Staphylococcus aureus. So if you ever hear about someone getting a staph infection, we actually have staph on our skin all the time. It's just something that we have there. But occasionally things get out of balance and the staph can then infect. If you have a cut or something like that, they can kind of, the thing that it's kind of strange to say, oh, if the body gets out of balance, if the bacteria get out of balance, but it is all about keeping this harmonious environment for everything to kind of thrive in equally. You don't want to have too much of one and not enough of another. It's all got to be balanced and monitored. We talked about antibiotic resistance before. So you get infection, antibiotics, and outcomes over here. Look at the chart and you can see what typically happens is that the bacteria that the antibiotics don't kill, they develop the resistance, and then those are the ones that replicate, and then you've got resistant bacteria. And because bacteria can replicate so quickly, this is a increasing problem. So the bacterial resistance on drugs, like I said, it evolves quickly. Stopping antibiotics early increases antibiotic resistance. So that's why they say make sure that you take all 10 days of this dose of antibiotics. If you're at day 5 and you're thinking, well, I'm feeling good, I'm just not going to finish these, you're really doing yourself a disservice, because what if there's a couple of resistant bacteria in there, you stop taking your antibiotics, and then boom, it comes back full force, and then you go to finish your antibiotics and they don't work. Then you have to switch antibiotics, and you just kind of create a bigger problem for yourself at that point. Okay, take a look at the chart there, and you can pause it if you need to. But if you, and also again, if you take them unnecessarily, that increases antibiotic resistance. So if you have a viral infection instead of a bacterial infection, you do not need antibiotics, because viruses won't be helped by taking those. It's for bacteria. Now we get into archaea. So they look physically similar to bacteria, but they are very genetically different. They have some features more similar to eukaryotes than they do to bacteria. And they tend to be like extremophiles, extreme heat, pressure, cold, salt, that sort of thing. Those are archaea. They can be difficult to study because they require those extreme conditions. I'm sure that you don't want to be like in a crater where it's, got lava all around you. You have to have that extreme environment, whatever it is, salt, cold, heat, pH, to study them. And you can take a look here at these. So these are the eukaryotic microbes. This is what they look like. So they've got things in here that are familiar: cytoplasm, lysosomes, your mitochondria, your ribosomes, your rough ER, plasma membranes, your cell coat. They have a nucleus, nucleolus. You have all of that, all the stuff that you're familiar with. So for eukaryotic cells, one of the biggest things is that you have the membrane-bound nucleus. It's got this protective layer around it. It has linear DNA. It has organelles. Everything has its little like pocket in the cell, and they all have individual functions. But the first eukaryotic cells were protists. Protists have evolved several different times in the phylogenetic tree. So you can take a look here. For eukarya, you have protists, and then plants, and then the plants go into fungi, and then the fungi to protists, and protists to animals. So you have these animal-like protists, fungi-like protists, and plant-like protists. They're not a monophyletic group, so you have, like I said, animal-like, plant-like, fungus-like, and then some of them don't even resemble any other eukaryotes, so they're kind of like off by themselves. For the animal-like protists, we consider those like heterotrophic. They eat food, hence animal-like. They're mobile, they can move around, and they hunt for prey. They move around, they look for things that they can eat. For fungus-like protists, again, those are heterotrophic. They eat food, but they're non-mobile. They don't move around, and they tend to grow in colonies. And for plant-like, these are autotrophic or photosynthetic. They make their own food. They're non-mobile. They can be single or multicellular. Protists, I switched to this one a little fast, but protists can be pathogenic. So protists can be parasitic. They have to live in a host organism for part of or all of their cycle. Like malaria, that's caused by a parasitic protozoan. Sickle cell anemia causes a change in the shape of the red blood cells, and then that makes them inhospitable to the protozoan. So if you've ever heard about those who carry malaria or frequently get malaria are actually like immune to sickle cell anemia because of this. And then here are some interesting links that you can take a look at. There's some films to watch. There's a TED Talk, one over fecal transplant in medicine. So you should definitely take a look at those. You may not be able to click on these links just by hovering over it. For me, I can, but you may have to just type them in. It's worth it. This is just part of the scientific process. So that ends what I have for chapter 17. Per usual, the actual chapter and the reading is going to give you a little bit more information than my PowerPoint overview. So definitely dive deep into the information that's in the chapters. It's going to give you a lot more. And if you are intrigued by something that is written in the chapter, by all means make a note, look it up, kind of follow your curiosity, because you'll probably learn something. Anyway, I'm going to stop this one here, and I will be back for chapter 18.